Good morning, ladies and gentlemen. Thank you for giving us the opportunity to introduce you to our activities in Duchenne muscular dystrophy. I will report on some efficacy data we have collected in the past years with our lead product, idebenone. We have trade names Catena and Raxone we use in different territories. In patients, older patients, not taking glucocorticoid steroids. Let me just introduce you what the company is doing. So we are a Swiss-based company with a focus on drug development for mitochondrial diseases or diseases that are influenced by mitochondria. You know that mitochondria are the organelles in the cells that provide energy. And we have currently our lead product, shown here to the left, in 3 indications. One is a prototype mitochondrial disease. It is an inherited form of blindness called Leber's hereditary optic neuropathy, on the top of the list. And here we are actually most advanced because the European Medicines Agency is currently evaluating our marketing authorization application for Europe in its final stage. And we expect the outcome of this evaluation very shortly. We also collaborate with the NIH, shown on the bottom, in another disease which is secondarily involved by mitochondrial pathology. That is primary progressive multiple sclerosis. And the NIH is investing in a long-term trial to assess the efficacy of this molecule in this particular form of MS. Now, for today, I would like to focus obviously on Duchenne muscular dystrophy, which also, as I will show in the later slide, has a mitochondrial component. And here I'm reporting the outcome of a phase 3 trial that we have concluded last year, and we are currently preparing for regulatory approval both in Europe and in the United States. So I'm showing you a picture of a young man with Duchenne muscular dystrophy. This is Robin. He lives in Zurich, in Switzerland, and illustrates the problem that I would like to discuss with you today, and that is respiratory illness associated with Duchenne muscular dystrophy. The progressive weakness of respiratory muscles leads to a restrictive pulmonary disease, which causes a lot of medical complications like ineffective cough, nocturnal hypoventilation, sleep disorder breathing, and ultimately daytime respiratory failure, as shown in this slide. We can measure the change in respiratory function with standard spirometry, shown here. You see in the center a so-called flow volume curve of a patient with Duchenne muscular dystrophy, and to the right in panel C, you see an age-matched healthy individual. Both individuals were 18 years when this was taken. And I'm grateful to Meier, Children's Hospital Philadelphia, who provided these figures. And you can see that in the so-called flow volume curve, there are 2 effects that are observed. First, the peak expiratory flow, PEF, that is the speed with which you can exhale air from the muscle, is reduced, as is the lung volume, forced vital capacity. It's also about half in the Duchenne patient compared to the healthy individual. And this is the medical problem I would like to take forward. As we all know, the introduction of glucocorticoid steroids and assisted ventilation regimes have greatly improved the life expectancy and the quality of life of patients with Duchenne muscular dystrophy. However, respiratory failure is still the leading cause of early morbidity and mortality in this disease. This is illustrated in a data set that was generated by the Children's Hospital Philadelphia, Meier and colleagues. And what it shows you that from age 10 onwards, roughly, there's a respiratory decline where the normalized peak expiratory flow and forced vital capacities are actually declining rapidly over the course of 10 years or so, and clearly below the expected normal rate of around 100%. So the dilemma that presents in all the patients is the following. It's a simplified graph, but I would like to use it to make my point. With increasing age, fewer patients can tolerate glucocorticoid steroids due to the known side effects. So initially, steroids are used, and they should be used because they're really beneficial and has been demonstrated in a lot of clinical trials. However, with age, side effects become a problem. And this is about the time when respiratory function declines rapidly in these patients about 10 years and older. And there's currently no treatment available in patients that are about at this location here. They cannot use steroids anymore because of side effect problems. They are sitting in a wheelchair at that time, most of them, and they experience rapid loss of respiratory function. So this is where we would like to position our our drug. Now, very brief and high level, I just would like to illustrate what the problem is. As other treatments that were introduced yesterday, like the NF-kappa-B approach and others, we are not addressing the lack of dystrophin. We are working on the downstream effect of the lack of dystrophin. And lack of dystrophin causes, amongst other things, that the muscle cell membrane becomes fragile. It gets ruptures during cycles of contraction and relaxation, leading to uncontrolled influx of calcium. Well, muscle can cope with calcium quite good, but at a certain point, this overload and homeostasis problem causes the fact that mitochondria become affected. And this subsequently causes an increased production of toxic reactive oxygen species called ROS and reduces energy production. Now, without going into the detailed biochemical mechanism of action, we have now evidence, and others have replicated this work, that idebenone actually can counteract the production of reactive oxygen species and can increase the output of cellular energy in situations where mitochondrial function is impaired. And that is important because it then can also counteract subsequent cell damage or even cell death. Now, we have invested for quite some time in a clinical development program that started in 2005 with a phase 2 trial that lasted between 2005 and 2007, which is published since quite some time, and now recently a phase 3 trial, what we call the DELOS trial, that also is recently published in The Lancet. I would like to give credit to the investigators who contributed to this work. I'm realizing that the format of my slides had a bit disrupted here, that the trial was really mainly run in Europe, but there were also US centers involved. European centers were in Belgium, France, Germany, Netherlands, Italy, Austria, and Switzerland, and Spain. So I would like to give credit to the investigators, but also would like to give credit and would like to thank the patients and the family who enrolled into this trial. Now, I'll give you just a high-level summary how the trial was set up. So patients 10 to 18 years were eligible to enroll. There was no selection status for the mutations that the patient can carry. They had to be off chronic steroids, and that's an important factor. So patients in the trial did not use steroids for at least 1 year prior to enrollment. Many of them actually did never use steroids before. And as a consequence of the age selection, there were 92% of the patients were non-ambulatory. We treated 31 patients with active drug, 33 were on placebo, and the average age was 14 years. Treatment was 12 months, and what I'm showing you here is what also is in the Lancet paper. That's a change from baseline. In gray, we see the placebo group, and in orange, always the active treatment group. And you see that, as expected, over a course of 12 months, the peak expiratory flow, as measured in the hospital setting, in the standard spirometry, declined by 8.8 percentage points. And this was clearly significant, but the decline in the active treatment group was much less. And there was a 71% relative reduction in this decline. So we could show that we could slow down the loss of respiratory function using peak expiratory flow normalized to patient age, as shown here. This was the primary endpoint. So progression in absolute values, if you look at just this peak expiratory flow in liters, is even more impressive from a trajectory. You see actually patients on placebo decline constantly, while actually patients on active remain stable. Now, we have also introduced a new device, which was actually very useful. It's a small handheld device, I have it here, which we gave out to the children and to the young men in the study and asked them to record every week, over 52 weeks, their respiratory function. And that is obviously very helpful because it generates a lot of data. If you look at the outcome of this analysis, as before, again, you see a decline almost linearly in the placebo group and a relative stabilization, which was statistically significant at week 52 in the active treatment group. Change in forced vital capacity. This is lung volume. That's important because I will show you later that change in lung volume is associated with early morbidity and mortality. Here we saw that in both groups, placebo and active, there's a decline, but the decline in the active treatment group was much slower than in the placebo group. Similarly, if you look at non-normalized data, just at not normalized to age and weight, this is just in liters, forced vital capacity, clear separation of the two lines. Now, this is a data set that shows all the summary data of primary and secondary endpoints together. And as I mentioned before, the study met its primary endpoint. In fact, I have to say that is the first phase 3 trial that in Duchenne muscular dystrophy was successful in the pre-specified primary endpoint analysis. And you see that in this summary data, you have the peak expiratory flow and forced vital capacity data. We looked at also forced expiratory volume in 1 second, another lung volume parameter. And you see that there's a treatment effect in favor of active treatment, Raxone Catena, for all of these analyses at month 6 and at month 12. A very consistent pattern of outcome. Now, I would like to address two things in the remaining part of my presentation. First, the study was quote-unquote, only 64 patients. So is the study robust enough to provide us with the evidence that we need? And second, I would like to address a couple of questions that are further away from the actual measurements. What is the impact of the observed changes for the patients? So this is an analysis where we investigated the robustness of the data set. That is the Monte Carlo simulation, where we have taken out 10% of all the data and run the analysis for the primary endpoint 1,000 times. So we took out randomly 6 patients, or 10% of the data, and just calculated what the effect size is. And you see clearly that in all of these 1,000 analyses, always the active treatment group is better than the placebo group. So that shows you that the trial itself, although small, is fairly robust. This is another way to show the similar information. That's a combined analysis of data from 35 patients in the placebo group and 35 patients in the active group, combining data from the phase 2 and the phase 3 trial. And that are so-called cumulative response plots. What you see here to the left is peak expiratory flow. And to the left of the zero line means patients have worsened in this parameter. To the right means that patients have improved. If you just look at the zero line, you see that 50% of the patients in the active treatment group showed some degree of worsening, but 75% in the placebo group actually showed a degree of worsening. If you go to 10%, so asking how many patients had a worsening by 10% or more, that was 12% in the active group and 40% in the placebo group. Importantly, the lines never touch or cross, and that is a sign that the data are fairly robust. Same pattern is seen with forced vital capacity. Now, you may ask, or you may have asked the question, I've just told you that the patients that were enrolled in the trial, some of them had used steroids before, and some of them had never used steroids before. In fact, about half of the patients never used steroids, and half of the patients did use. Question is, do both patient populations benefit equally from treatment? And the answer is yes. So far as we can see is the following. This is the PEF analysis and forced vital capacity. That is speed of air, and this is lung volumes. And these are the effect sizes observed for patients who never took steroids and those who have previously taken steroids but have washed out before the trial. And you see the effect sizes are fairly similar for both parameters. From that we conclude that the treatment effect in favor of Raxone is not influenced by previous steroid use, although at the time of the trial, patients did not take steroids. That's another pre-specified analysis. We looked at patients that had a reduction in peak cough flow to 160 liters per minute at any time point during the trial. Why is that important? It is important because if you fall below 160 liters per minute in peak cough flow, then you can no longer effectively clear the airways from mucus, which increases the risk of airway infection. And that is very well established in the literature. So we had, in the beginning of the trial, we had 26 patients who had at baseline above 160 liters per minute in the active treatment group and 33 in the placebo group. You see that there were more patients in the placebo group who fell below this clinically relevant threshold of 160 liters per minute in peak cough flow, indicating again the benefit that Raxone treatment exerted. Another way to look at the data is forced vital capacity. There's ample of evidence that shows that if the forced vital capacity, the lung volume in a patient falls below 1 liter, that is associated with early mortality the 5-year average survival time in patients with less than 1 liter forced vital capacity is only 8%. So we had 6 patients in the active treatment group and 9 patients in the placebo group who had between 1 and 1.5 liters at baseline. These patients were at risk of losing their forced vital capacity to the critical threshold of 1 liter during 12-month study. And in fact, there was only 1 on the active treatment group, but 5 on the placebo group who crossed that clinically relevant threshold. That's an analysis where we look at infections, and we have done it in the following way. We have blinded the reports of upper and lower respiratory tract infections to 2 experts in the field. Both individuals were ex-FDA people. One was a senior representative of the Division of Infectious Diseases, and the other one was a senior representative of the Division of Pulmonology. And we asked them to classify diseases, airway diseases like infections, which they consider as medically severe or relevant for patients with DMD. Then they gave us a list of diseases they were looking at, and then we unblinded. And we can clearly demonstrate that there were more patients, clearly more patients in the placebo group compared to the active treatment group who experienced these clinically relevant airway infections. There were 30 events reported in placebo and only 7 in the active treatment group. And if you count the number of days patients reported having one of these severe infections, it was 231 in the placebo group, but only 82 in the active treatment group. Now, we gave them, the same experts, the task to also classify the use of antibiotics, which are typically associated with treatment of airway infections. So they came back to us with a list of antibiotics that are typically used to treat airway infections. And then we counted the number of patients and the number of days these antibiotics were used. You clearly see that there's a higher number of patients in the placebo group, 14 against 8, compared to the active treatment group that actually used these antibiotics. But what I think is very interesting is that the number of days, total days reported for antibiotic use in the placebo group was almost twice as large as in the active treatment group. So we can translate the observation that we have made with the respiratory function tests into clinically relevant information, such as the number of airway infections and the use of antibiotics. And I think that is putting the entire outcome of the trial into a medical context. I'd like to summarize the data that we have shown. There is, as I just showed you, a sizable proportion of patients who cannot tolerate steroids at the time when respiratory loss becomes evident. And the recent study by Synergy indicated that about 40% of patients in the United States, 10 years and older, are not using steroids due to side effects. There's an urgent medical need to slow down the decline of respiratory function in these patients. And the available data from our investigational drug indicates that we can slow down the loss of respiratory function in patients not using steroids. Raxone was tested in patients without restriction of any mutational or disease status, ambulatory, non-ambulatory, or irrespective of mutation. And I should say that Raxone is an oral medication. It's given 3 times a day, and we use it 2 tablets. With this, I would like to thank you for your attention.